Good morning, everyone. It is my pleasure to address you here today, and I would like to thank the organizers for inviting me to attending this very exciting meeting. The 30 years after the HIV discovery, we still do not have a cure for HIV. However, the impressive research has been recently discovered that delivered to maybe we can cure the HIV, or we can we can control the HIV for a long period of time. So today I would like to present some of the advanced research in HIV research area. HIV is HIV pass infected into the host cell by the. Can I have a? Okay. HIV infected to the host cell by binding of GP120 to the host receptor CD4, and then generated the cDNA by reverse transcriptase, and then integrate into the chromosomal DNA, and then transcribe the progeny DNA and generate the progeny virus. In the process of the HIV, the life cycle, that highly active antiretroviral virus targeting each steps. And here is showing that the 4 different kind of the inhibitors, and that is the entry inhibitors and NRTI, the nucleoside reverse transcriptase inhibitor, or integrase inhibitor, or protease inhibitors. And this ALT is around like 20 different kind of them. And this limited number of the inhibitors are one day, if we encounter the mutant virus, then we have a problem. So therefore, supportive that the therapeutics is definitely required. Here is one of the examples that the heart treatment can reduce the viremia, as showing on the right side. However, without heart, then maybe that the human life is wiped out by today. So we have to appreciate of the ALT. And the point that I want to mention in here is that although that heart is very effective controlling HIV viremia, however, there are still that 50 copies per plasma is still present, and those proviruses continuously replicate the virus and generate the mutant virus sometimes, and continuously replicate inside of the host cell. So therefore, this around 20 different kind of number of the ALT may not be useful one day. So recently scientists are working hard enough to generate the supportive therapeutics. And here is the list of the recent advanced research that has been done lately. And one of the important thing is that use of broadly neutralizing antibody. And the other thing is conventionally we believed that the CD4 is the reservoir. However, it recently found out that the hematopoietic stem cell also be one of the reservoir too. And the stem cell replacement can be used for the alternative therapy for the HIV eradication. And the other thing is the gene therapy, using a zinc finger nuclease, and the RNA therapy can be used to treat the HIV. And when the HIV infected the host cell, that HIV used a lot of host factors to replicate it. And maybe that is another alternative target instead of a viral gene. So that is another way that we should go. And the other thing is that although that we can control the HIV very well with the heart therapy, however, is there any way that we can eliminate those latency virus? So I will briefly talk about each topics in this talk and in the next slide. So broadly neutralizing antibody. My major is the T-cell immunology. I have studied for 5 years at the Judy Lieberman's lab for how to elicit cell-mediated immunity and the T-cell immunity. And there's $100 billion has been spent to elicit the T-cell immune response and the cell-mediated immune response. However, after the paper that has been published by Kwang and the Muscular Nusanjui group, that shows that the broadly neutralizing antibody was sufficient enough to neutralize more than 90% of HIV. So is this the way that we should go? So what they're showing is that they are able to find the broadly neutralizing antibody that targeting to the conserved region of the CD4. And these broadly neutralizing antibody capable of blocking large quantity of many different strains of HIV. And this clone now is making this clone, based on this clone, now that the scientists made a recombinant antibody, and that is in a clinical trial 1-2. And this broadly neutralizing antibody and bind to that GP120, where actually bind to CD4 molecule. So this CD4 binding site of the HIV is very conserved among the different strains of virus. And this antibody is usually present in the elite controllers, like long-term non-progressors. And however, this titer is not strong enough, but with a small titer, they are able to control the HIV. And on the right slide, we have another broadly neutralizing antibody that binds to that 4E10 and 2F5, which is the GP41 site. And that also gives us the hope that those kind of 7 different kind of broadly neutralizing antibody right now has been identified. And we have a big chance to neutralize most of the virus that is present in this time point. So using a broadly neutralizing antibody is another way that we can develop supportive therapy for HIV. And the other finding in recently that conventionally we believe that CD4 is the reservoir for the HIV. However, that the Collins group, they find out that the hematopoietic stem cell is infectable with HIV. So usually that's X5 virus, which use the CCL5 as a co-receptor that transmitting and initiating the infection. However, that HIV progress, then CCL5, that the X5 virus converting to the X4 virus, which is infected to the T cell rapidly. And those viruses are the ones that are able to infect the hematopoietic stem cells. So thus, we have a question. So then, right now, all the development or HIV research is focusing on the how do we remove the latent HIV in T cell and CD4 T cell. Now we have another barrier that we have to eliminate the virus from the hematopoietic stem cell. So then is there any alternative way to overcome this problem? Yes, it is. So we have easiest way is that change the whole stem cell therapy, replacement of stem cell from normal stem cell to the normal stem cell replacements from the HIV infected patients. And one of the astonishing discovery lately that we have is that this Mr. Brown's syndrome. So it's called Brown syndrome. So this patient is like a celebrity right now. He's more famous than doctors right now. He go to the many places, give a talk how he has been cured. And this patient is a 40 years old male and diagnosed with HIV for 10 years and heart for 4 years. And somehow he has acute myeloid leukemia, so he need to transplant to his hematopoietic stem cell. So the Dr. Hutter have decided he know that CCL5 deleted mutant, that homozygous, that bone marrow is resistant to HIV infection. So he was looking for the CCL5 delta mutant homozygous donors and then transplant his bone marrow to this patient. And then after the 4 years that there's no viral has been detected in this patient. So what is the CCL5? So as I mentioned earlier, that the initial transmission and HIV infection is mediated by CCL5 trophic, the X5 trophic HIV. However, that what we knew is that the CCL5, that normal CCL5 that present in the 81% of a Caucasian population usually get infected HIV, progress to AIDS normally. On the other hand, that the homozygous that deleted mutant delta CCL532 that present in 1% Caucasian, they are very resistant for HIV infection. So thus, based on this finding that maybe elimination of CCL5 would not affect our human immunity at all, but its benefit for HIV resistant. So what Dr. Hutter did is that he find the bone marrow donor, which is HIV negative, and then HLA matched to the Mr. Brown, and then he transplant bone marrow to this Mr. Brown. And what they found is that after the nearly almost 100% of the blood cells is a CCL5 negative, and the heart was discontinued after bone marrow transplantation, and luckily that undetectable viral load 4 years after transplantation. And this is the first case that complete eradication or cure of the HIV. One of the problems of using this therapy is that finding a stem cell has a homozygous delta 32 is not easy. So is there any alternative way that we should go? So yes, this might be the way. So there's two ways that we can do is gene therapy. One is using RNA interference. RNA interference has been discovered by Andrew Fire and Craig Rumello, and they won the Nobel Prize in 2006. And so when I was in a Harvard Medical School, I was one of the scientists that shows that the RNA can inhibit the HIV, that has been published in 2002 in the Nature Medicine, and in collaboration with Phil Sharp in MIT and with Dr. Lieberman and Dr. Shankar, and also one of the leading scientists is John Rashi and Ben Burkhout are the one that who using RNA interference as a supportive therapy in HIV. So what we can use is that RNA interference is mediated by small interfering RNA, that 19 to 23 nucleotide. Once we add to the cell, and those sRNA will complex with the in host protein is called RISC, and then they find the target mRNA and degrade the mRNA. So, however, transfection into the primary cell is very difficult. So, another approach that we can do is that make a plasmid DNA to generate the short hairpin RNA, which is the